You guys can see my PowerPoint, correct? Someone give me a hell yes.
Yes, sir.
Yeah, we can
Okay.
see it.
I'll never get a hell yes. Yes, sir. That's very respectful. Thank you. What can we look at? We're not going to go over photosynthesis again. We went over the Z scheme this last time. Only activate my laser pointer if I go into presentation mode. So we talked about the Z scheme before, right? We have a lot of light that's harvested into this one concentrated molecule center, photosystem 2. So light energizes the electrons from water. Water is oxidized. These high energy electrons are then tumbling down in energy, and as they tumble down in energy, they create a proton motive force here, cyclic, and then this energy, this electron, and when it's a lower energy, is captured by another photosystem called P700. Light energizes that to a high energy electron again, and then as it tumbles down, more proton gradient, more ATP, and then NADPH is produced. So from this light cycle, we produce NADPH and we produced ATP. And then from that, we then, oh, isn't this nice? So this is just a complicated electron transport scheme for this. So here you can see light enters, boom, and then water is split, and then the electrons flow. You don't have to memorize this. You just need to look at this guy. The PQH2. It's just the scientific dorky saying of quinone. Remember quinone and electron transport chain? Same guy. So it's a very popular intermediate. So we split water, electrons go through electron transport chain. That's all you have to know, right? And then it goes through the electron transport chain, and electrons are ultimate electron acceptor for the light. This is a good exam question. What is the ultimate electron acceptor for oxic photosynthesis? It's NADP. And then, so here in the stroma, we have NADPH and ATP production. And then we don't need to memorize all this, but this is a sugar. And this is a gas of CO2, a gas molecule of CO2. This gas is added to this sugar. And now what we've done is we've incorporated carbon that was in the atmosphere, inaccessible to the biosphere. No organism can handle it, deal with it, work with it, incorporate it, and love it. So here is where plants really, and phototrophs really, they are the beginning of our, carbon cycle, where CO2 is incorporated into a molecule. And then from here, all these steps are just, look at this, we add ATP, we add NADPH, we just basically turn the CO2 into a CH2. And then once we're at ribulose bisphosphate, we can start to rationalize, getting into glucose or nucleotides or whatever. So that's just a review. That's the overall stoichiometry is what I would pay attention to on this slide. Okay, figure 1418, I just care about that. We went through CO2 entering Rubisco. We go through the cycle. Then we have fructose for biosynthesis. We have to generate some of the molecules and retain it so that we can go here. But for the most part, we can think of CO2 plus ATP plus NADPH equals glucose, fructose, whatever. And that's all given here. Okay, so that's called the Calvin cycle. That's called the dark reaction. It can occur in the light, as was brought out last time, but that's the Calvin cycle. So we have the light cycle and the dark cycle. We have the Calvin cycle, which is the dark cycle. We talked about reverse TCA, and all we really need to know here, we're not going to try to commit to memory all of these things. The point of this concept, reverse TCA, is that we did cover this, right? Yes, right? Yes, reverse TCA. So the point of a reverse TCA is that CO2 is being incorporated here, right? And so you have plants that don't necessarily use the Calvin cycle. They can use the reverse TCA. You just need something from which you can take CO2 out of the air and create sugars out of it. And so here you can see reverse TCA. We see a lot of these metabolites that are very familiar. They're in the TCA cycle. We reverse this, And then by incorporating CO2 where we were producing CO2 before, when we were learning about glycolysis and aerobic respiration, here, if you reverse everything, then you're going to see CO2 enter these pathways, and then you're going to come up with glyceraldehyde 3-phosphate, and then you can get to your cell material. So the bottom line here is from photosynthesis and from this reverse TCA is these are ways from which an organism that doesn't have a diet, a carbon-based diet, meaning it doesn't take any carbon in through its diet. Plants don't take carbon in their diet, meaning in the soil, they're not taking in glucose, they're not taking in acetate, they're not taking in nucleotides, no carbon-based molecules are coming in through their roots. All their carbon is coming in through the air. So that makes them autotrophic, right? Here we have the same concept where CO2 is being incorporated out of the air. So we have a mechanism of living independent of a carbon-based diet. We don't need to take any carbon in there. Okay. So anytime you see a metabolism where the electron donor is not a carbon-based, that means that, oh, well, this thing oxidizes iron and it reduces sulfate. Where's the carbon? It has to then have some mechanism to incorporate carbon. Right away in your mind, when you see that chemolithotrophic stuff, not the chemo organotrophs like us, we take in organic molecules. Our carbon-based molecules come from those carbons, right? We turn our glucose into nucleotides, sugars, fats, anything we want. But for things that don't have a carbon-based diet required, where you say, okay, this thing oxidizes iron and it reduces sulfate. Where's the carbon? You know right away. It must have some mechanism of converting CO2 into biomass. A lot of times that'll either be the Calvin cycle or the reverse TCA cycle. These processes are not um, only for uh, phototrophic microbes. Okay? Calvin cycle and reverse TCA also exists for chemolithotrophs that don't utilize sunlight as an energy source. That was a lot. Okay. I think that mostly came out okay. So you learned about nitrogenase. I'm going to keep this um, respiration. So I think, talked about this slide, and this is just an order of different things that can be reduced and what the energy levels of them are. And so the higher up, this is an important thing to distinguish on figure 14.25. This is a list of molecules that can be electron acceptors. That's what this is. This is a list of electron acceptors, 14.25. And so you can see it right here, anaerobic respiration. And here you can see energy. So when you're dealing with trying to guess what is the best electron acceptor, you always want to be low, okay? Oxygen should be a good reference point for you to be able to understand how to utilize this. It's definitely going to be on the final. And so I'm going to give you this information. I'm going to ask you to use it. So what's the worst electron acceptor on this chart? Anybody want to pipe up? So worst electron acceptor on this figure.
Hydrogen.
Hydrogen. Okay, very good. Hydrogen. So I want you to pay attention to that because I'm going to be going over this in a minute. Imagine that. So if you're a hydrogen producer, man, you are just living on the edge. You're barely able to squeak out an ATP. You probably, you know what, you can't justify it. We'll talk about how this happens. But for the most part, you can judge the value of an electron acceptor by assessing what its redox potential is. Its redox potential is an indication of how good of an electron acceptor all of these components are. Okay. So give me a second. I have to organize my thoughts. Okay. Thoughts organized. Um, so let me see what slide is next. want you to pay attention to the redox couple of these different electron donors. It's difficult. Sorry, the reason why I'm collecting my thoughts, I'm trying to consolidate information that I usually would spend more time giving you, and I'm trying to give you information um, that is more. It's just more summarized. So I have to make sure that I'm careful with my words. Don't worry, I'm not going to curse. Okay, so iron bacteria. So these are all groups of chemolithotrophs. See that? And we have varying levels of delta Gs. Okay. And so what this says is that this is an indication of the quality of the electron acceptor and the electron donor. So that's the redox couple. When you minus the electron donor with the electron acceptor, you get a difference in voltage. Really, we don't care about voltage. We care about delta Gs. And so here is the delta Gs for those couples. So that's the best way to kind of read this whole thing. You guys know about aerobic and anaerobic respiration. And so this slide just basically is a summary of everything I just said. You'll have to read it. It reads more technically, but that's basically all this stuff is. Lithotrophs conserve energy from oxidation of inorganic molecules. I just went over all this, hydrogenase and energetics of H2 oxidation. So now I want you to slow down and take it easy, man. Take a breath. All right. This is where you guys can see me. Yes. All right. That should have been all review for the most part. This next 10 minutes kept me up all night. All right. Oh no. Boy, I would have gotten no sleep if I knew I was going to have a marker that didn't work. Mayday, mayday. Oh, there's a marker on the floor over here. Excuse me. And if this one doesn't work, I might freak out. All right, chemolithotrophs. I know you were looking forward to me flipping out. Oh well. I'm not going to define chemolithotrophs. Chemolithotrophs are organisms, microbes that oxidize inorganic molecules as an energy source, electron donor. If you can have chemolithotrophs that are aerobic or anaerobic, that just depends on the electron acceptor. So we have hydrogen oxidation, right? And here we have H2 plus half of O2, and this goes to water. This would be an organism that oxidizes hydrogen, and the delta G here is going to be minus 237 kilojoules per mole. So it's a highly beneficial microbe. At the center of this is an enzyme called hydrogenase. Hydrogenase enzyme is ubiquitous in microbial metabolisms. It's a really key enzyme that exists in a lot of microbes that allow a lot of microbes to work together. Hydrogenase catalyzes the interconversion of hydrogen gas and protons, right? And so, let me write that a little bit better. Jesus. I'm trying to be mindful that this is somewhat hard to read. Okay. And so this enzyme is really important. And if you have hydrogen oxidizing capabilities, it really helps out any microbe that has hydrogen or protons as a part of their metabolism. So let me give you an example. Before I do that, let me just see what the next slides are. Okay, before we go that H2 bacteria, if you have something that does that, all right, then conceding that that your electron donor is not organic, and that you need to you need to fix your carbon yourself. Okay, and so that means that you're going to require a way to do this. Now, these hydrogen molecules, these autotrophs, I should say, they fix CO2 by the Calvin cycle. So it's not just a plant thing. Okay, and so you want to remember that every time that we have something inorganic as our electron donor, that we're going to need to use either the Calvin cycle or reverse TCA in order to fix our carbon. Here's where these guys are really useful. Say that you have an acetogen and they take acetate, and then they'll produce H2 plus CH4, for instance. Okay, I'm sorry. This is a methanogen. Acetogen produces this, and so it'll take propionate, and that'll produce acetate. Okay. So here we have an H2 production. Now, H2 bacteria consume H2. Methanogens have hydrogenase, though, right? Because you can see, we'll put our hydrogenase back up here. Okay, and our hydrogenase interconverts this. So anytime you see hydrogen and protons being interexchanged, you have a hydrogenase here. So since we're making a H2, we can say that hydrogenase is going in this direction. Okay, and so this is enabling the microbe to commit to a certain metabolism. In this case, and this is the bottom line, the delta G here is positive, delta G here is negative. It's a really key concept here. And so an acetogen got no business to live. How are you going to make that's sloppy? You committed a whole metabolism for a positive delta G couple? That's silliness. Well, we need this, right? Because organic molecules are breaking down in the soil, and we start off with like 12, 14 carbon molecules. All of our complicated stuff, all our biostructures have all these carbons bound. But, you know, during the big sleep, when I'm committing my, mortal remains to the 6 feet under situation, I need to be able to break down and give back all the carbon that I took from the atmosphere during my time here. And all the animals do, all the plants do. You got to give it back. It's not your carbon. It's not your nitrogen. It's not your carbon, sulfur, oxygen, none of that stuff. It's all, you got to give it back. How are you going to give it up? Microbes break it down into simpler carbon units, right? We go from 14s to 6s to threes. It's a 3-carbon molecule propionate. There's a 2-carbon molecule in acetate. Yes, you technically are giving off plus CO2, but this is a positive delta G. So we have an integral uh, player in organic breakdown that has a positive delta G. But because a methanogen is around, and methanogens have this hydrogenase, they can then take the electrons oxidized from acetate, combine them with protons to make hydrogen. When you combine, so then you can really add, you can treat this like two independent reactions. You're going to say, okay, cetogen has no business to live. Methanogens are doing great. Problem is, methanogens needs acetate to live. If you combine the delta G's here and here, and if you get a net delta G that's negative, then these microbes will work together. And the center of all this is hydrogenase. Hydrogenase is what makes it all work. I can give you a million examples of how hydrogen is a big player in a lot of metabolisms. A lot of times microbes, if you were to think about it as something they're trying to figure out, you can almost make a living off of anything as long as you can make H2 or H plus as a part of what either is being consumed or produced. Then you can hook up your metabolism into a methanogen, and the methanogen will make it happen. Gotta stay anaerobic, kind of sucks, but still. So, any questions on that? That's the main concept I wanted to offer here.
Yeah, I have a question. So you said hydrogenase is gonna make things work, right?
Yes.
And so that reaction above is gonna be spontaneous and it coupling with
Yes.
wait, it's supposed to be non-spontaneous because the hydrogen is really high up on the redox potential. So it's have a harder time of accepting electron.
Yeah, that's why anyone in the situation of dealing with the hydrogen is usually good, but the methanogens can make it work.
Oh.
In this case, hydrogen is not the electron acceptor. Okay, so um, it's H plus that's the electron acceptor. You're thinking about it a little bit opposite, but yes, but you're absolutely right. It's counterintuitive.
Yeah, so the important thing is hydrogenase has to be coupled with methan, methanogen.
Methanogens. Here's
Mhm.
another way to think about it.
It's supposed to be non-spontaneous if H has a harder time of accepting electron.
Here, it's not accepting electrons; it's being produced.
Yeah, I'm
It's
talking
flipping
about
the delta G.
hydrogenase. Yeah,
Like when you reverse the reaction, you flip the delta G.
okay.
Becomes favorable in that situation. When it's not an acceptor, if it's a donor, you know, that's something that's tricky about that chart. That's why I said you have to remember that you're dealing with electron acceptors. H plus is the electron acceptor there. H2 is produced, and so I think that what you're trying to say is that because H plus, if you look at that chart, H plus being an electron acceptor produces H2 becomes a very bad electron acceptor. Correct? That's your basic dilemma.
Yeah.
Okay. And so, and that's a true statement. True
Mhm.
statement. The antigens can make it work. The antigens can make it work.
Mhm.
And so their delta G is negative. They are growing really slow, but they're the terminal part of all organic breakdown. And so because they can make it work, a lot of microbes that are dealing with this hook up with methanogens. Because methanogens can make it work. As a concept, the lower that you go down in the soil, the higher the energy of the molecules, the harder it is to find electron acceptor. All right, let's move on. I don't want to stay with this too much, because most of this stuff is not going to be emphasized. Okay, and so that is just an example of how hydrogen. Oh wait, let me share my screen. Well, I'm not gonna share my screen yet. Let me just finish this. The book really is. This is the hardest part of microbiology to teach. This particular topic, this part of the course could be a course in and of itself. I could split this part of the course into several courses. And so it's really hard for students to understand it because it's not really the right way to teach it, but it's an appropriate introduction into all these topics. So I agree with this. And so here we have an example of sulfur. So when we started all this, I wanted to talk about geochemistry, biogeochemistry. And so here we see H2S being converted to sulfate, okay, plus 2 H pluses. So now on this end, hydrogenase can be on some other end dragging this to H2. Every time now that you see this, you know another microbe really can work with this. Okay. We have another example of a reaction. S2O3, 2 minus, goes to, I'm sorry. I think I wrote this wrong in my notes. I'm pretty sure there has to be an oxygen here. Sulfate. So the main thing here is that sulfur is cycling between different forms. Okay, we have hydrogen sulfide, we have thiosulfate, we have sulfate over here. And so sulfate, here, let's put some fun facts here. Sulfate, if you guys haven't caught on yet, sulfate is the most common final product for sulfur bacteria. Sulfate is being produced here. Okay. Sulfate also happens, sulfate, very popular electron acceptor. So that works out for everybody. And so I've said this a million times. If you're at the marsh side of the marshes and you smell that kind of rotten egg smell, what you're smelling is when sulfate is an electron acceptor. You can add 2 protons here too. When sulfate is an electron acceptor, then you produce hydrogen gas. And when you produce hydrogen gas, then you get the H2S oxidizers a chance to make a living. And so the most important thing from this whole slide is like, because I know this can seem like, why the hell are we talking about this? The bottom line here that you should notice and what is to be appreciated is that we are cycling sulfur between different forms of sulfur, and that microbes are one cycling this. And this would be the cycle of sulfur in the biosphere. It's the most important thing. I can go on and on. I will not. That's just the most important thing, is that sulfur is interchanging between different forms. We have iron oxidation. Iron, and this is important because iron is in our bodies, right? It's on all hemoglobin. And so we need to have, whenever the biosphere is demanding a certain element be in a certain form, uh, it needs to be available. So the bioavailability is going to rely on microbes ensuring that these forms of the iron are in balance with the needs of the biosphere. Makes sense. Can't get locked up all in one form. There has to be a way to bring it back. We can't depend upon nature to put it back in the form that we need. We need a way to make it available, both in 3+ and 2+. So iron is a part of all the biosphere and all the organisms in the biosphere. It implies that bacteria have found a way to balance the availability of these different forms of iron. So we'll talk about iron oxidation. Iron oxidation is interesting because it involves some tricky metabolism. We'll go over that in a minute. I'll need the PowerPoints though. We won't convert yet. So we make NAD to NADH, and the different interesting thing here is that the delta G is positive. Super weird. So now we have iron 3+, okay? And then what happens is, as this goes into the ferric hydroxide, this is a delta G negative process. So this drives the whole thing, kind of pulls everything in that direction. And so, and obviously there's 3 waters here. I don't feel like drawing them, okay? So formation of the ferric hydroxide pulls iron oxidation favorably. So, and this is probably the last that I'll talk about iron. Electrons from iron 2+ reduce NAD+ to make NAG H and oxygen. NAD formation, NADH formation, and ATP from O2 to sugars. Here, NADH formation and ATP from oxygen to water, because oxygen is an electron acceptor, make sugars via Calvin cycle. Another example of the Calvin cycle not being in a phototroph. So in other words, the NADH and the ATP, this is another way of saying that NADH formation and ATP formation from respiration drives the energy that converts CO2 to sugars. In photosynthesis, we know that light energy drove the synthesis of NADPH and ATP. And then we were able to take CO2 with that NADPH and ATP and make sugars. Here's an example of oxidizing a molecule. Instead of receiving that from the sunlight, we're using the energy from an electron donor. That energy used to make ATP and NADH. When we can couple NADH and ATP with CO2, then we make sugars. That's the fascinating thing about the iron oxidation, is that we have a way to fix CO2 in the absence of sunlight. It's a good example. There's many examples. It's the example I'm going to hang on to. Okay. Any questions here? Let me share my slide. I'm going to change this.
I have a side question.
What's the product?
Go ahead.
Go ahead. Let's switch. Let me share my screen. Uh-oh. Look at all that rust. Isn't that beautiful? So that's that reaction going on. Hydroxide. So you create rust, basically. Can you now ask your question? What's the product? Is that what you're asking? Here? I don't know if I can mute you guys. What's going on?
We can hear you.
Oh, okay. And I can hear you. I thought it was a question. So let's look at this. So this is just a gram-negative bacteria that is an iron oxidizer. And so here we see that clearly electrons come from iron. And then the electrons are transported to this rustocyanin. This rustocyanin basically offers two options for the electrons. It just routes electrons in both directions. One is an electron acceptor in oxygen. So this is going to form ATP. We know that this is a proton motive force. The other electron has to go up its energy, so it's actually going to go upwards. And this is going to be reverse energy electron flow to make NADH, and eventually we're going to use the Calvin cycle to make glucose. So I'm going to amend this talk to not include any. Yes, go ahead.
Is it double membrane?
Yeah, this is yes, that's right.
So it's gram negative? No.
That's right, it's gram negative, that's right.
Wait, but it's a bacteria. Oh yeah, gram negative bacteria.
Gram negative bacteria.
Cool.
So I know this is a little bit off. This stuff really, when I was a microbiology student, this stuff is super difficult. If you're not interested in this stuff, it's hard to contextualize it. I would classify myself as not interested back then. I became fascinated with it later, but it takes a lot of exposure in order to really appreciate it. I think the best thing to understand from this is that this is just a scheme to interconvert iron, and that matters for the biosphere. I can do this trick, and I have in my notes. I have nitrogen and how to the nitrogen cycle. I'm not going to cover it. I'm not going to cover it. I think you guys have learned enough about nitrogen that you can appreciate how nitrogen cycles. I can see all of you out there nod your head. Oh yeah, we get it. Let's move on. Okay, we'll move on. I'm cutting this topic short. I gave examples in nitrogen, iron, and sulfur cycling in the biosphere into different forms using the using microbes. So the microbes are cycling elements in different forms, and these different forms can be used by different players in the biosphere, different members, different species. I also talked about how metabolisms can be linked so that the products of one microbe can be the substrates for another, and that depending on how it's situated, we can have a whole string. In theory, I should be able to you guys can still see me, or you still see my PowerPoint?
We can see you.
Yeah. So if I have a process that's delta G positive, delta G positive, delta G positive, and over here I have a delta G negative, and we have one metabolism that can string all of them over. Okay? So they're linked like this. So microbial communities are essential. Those are the themes. The moment we all been waiting for. Microbial infection and pathogenesis. Oh man, this is good. All right, I'm sharing my screen. Oh, wait, let me get to the right PowerPoint. I'm gonna have to ask you guys. Seems unfair. Let me. You guys don't know what the hell I'm talking about. Let me show you what's unfair. It's not that unfair. Share my screen. You guys see chapter 24? Okay, so I'm gonna ask chapter 24 to be on you. This is actually low barrier. This is actually easy stuff. All right. It's just talking about the different habitats and portal of entries for a lot of different microbes. And we all get inoculated and infected with microbes when we're born. And our urogenital tract, our ears, our nose, um, the intestinal tract wall cavity, our eyes, everything the skin, everything that's exposed to the atmosphere is basically inoculated. And in order to have a healthy microbiome, you need to have been exposed to healthy microbiome. So usually this happens at birth, and usually, mommy takes care of that in the womb. Thank you, mom, for all your bacteria. I appreciate it. And so, it's so weird. I would have done a different job of presenting. Don't worry, ladies. There'll be a picture of a guy later. All right. So here, all they're pointing out is the different routes, all these little places where bacteria can live. The teeth are a really major one because that's really um, close to a blood supply. So a lot of times that's a portal of entry for a lot of bad stuff. So I'm just going to leave it up to you to kind of read about this. This is very accessible information. You can go through your PowerPoints and kind of go, what do I care about? Just reading this stuff, and it's just easy. Don't worry about this stuff. Don't memorize, not that you were ever going to, all of the bacteria. The only point of this slide is that as we go from the esophagus through the digestive system all the way to the anus, that there is a difference in pH. We go from a pH 2 to a pH 7. And the intestines are going to have different a pH gradient, and they're also going to have a macromolecule presentation gradient. It means that things that are in the stomach are not going to be present in the appendix and the anus and stuff like that. So you're going to have different habitats for different microbes, and you're going to populate different parts of the intestines with different bacteria. Okay, and that's the main point here. And all the secretion of acid, macromolecules, amino acids, fatty acids, I will be holding you to that. Okay? So pH gradient, difference on where things are absorbed. Give a, and you give praise to the bacteria along the way. You don't have to memorize the bacteria. And so then you're going to just read through this, and you're going to just make sense of this. I don't want to go through it. I don't think it's a good use of our time because I think it's all fairly accessible. Saliva contains antimicrobial enzymes, so do your tears. Um, Otherwise, you have your skin has a low pH that is inhibitory to a lot of microbial growth. It's also covered with a bunch of microbes that inhibit that serves as a shield for you. That's why you don't want to scrub too much, because if you scrub all the bacteria off, you're not only going to irritate your skin, but you allow for a portal of entry for microbes that are not welcome. So I'm going to just kind of leave this up to you. There you go, ladies. Okay. So um, the vaginal tract is obviously a portal of entry, and so there's going to be conditions that um, um, discourage bacteria from um, colonizing that, and the same thing is true for the penis. Okay. And yeah, so I'll let you guys just read all that. It's pretty accessible, and mostly I just want to. I think I've talked about the obesity thing. Obesity is more prevalent in some bacteria. So this obese mice with the lean mice, the difference is the efficiency from which they're fermenting. Remember I told you that before? This is where it really plays in. So um, this obese mouse has the same diet, but the difference is it's more efficient at converting the biomass into the food into biomass in this terminal step. It's got a higher percentage of methanogens, basically. That's what this slide is really showing you. Okay, so I'm going to leave that up to you. You guys see my PowerPoint still? Different PowerPoint? This PowerPoint?
We can see it.
Yeah.
All right, cool. All right. Sorry to rush through that. It's accessible. I don't think it's a good use of my time to explain things that you could read just fine. So read it, know it, love it. And it's basically a story of of the normal microflora that's on your body, throughout your body. Now that you're experts on how the body works, now let's talk about disease. That's how medical school works. They teach you how everything works, and then teach you how everything breaks, and then teach you how to treat it, and then they put you out there. So that was med school. How was it? Okay. So we have different stages of, that's not what I want. I want a laser pointer. Let me just really quick. You guys see me? All right, let me just start it off appropriately. And so we're going to have 25.1 infection. I know the presentation thus far has been really rushed. Going at normal Dr. Cruz speed now. I just want to get through that material. This stuff's good. And I wanted to offer it to you, and I think that this is what you guys want to see. So infection, we have to define infection. So this is a growth of a microorganism in a host. Growth of a microorganism. We're going to have to come up with MOO. It's an acronym for microorganism. It's too many letters, so we're going to say microorganism. So from now on, when I put MOO, that's what it means. In or on a host. Okay. So that's the an important concept. Does not mean disease. Does not mean disease. So microbial pathogenesis occurs in steps. And so here we have all the steps for typical pathogens. First, you have to be exposed to the pathogen. And then they have to be able to bind. They can't bind, they can't engage, and they can't certainly get in the cell. So there's adherence, right? And then they have to, at some point, traverse across the barrier in order to access the cell. This is pathogenesis. This is not symbiosis. Okay? And so then we have invasion. Okay, and then when you have invasion, you multiply in the tissue. And then from here, you can go in one or two directions. Usually you go in both. You further grow and you get beyond your point of original infection and you go into other places. You're spreading. This causes tissue or systematic damage, right? You had a microbe, it went into one cell, it grew in that cell, blew it up. Now there's a lot of microbes. Now it goes into neighboring cells. It's spreading and it's killing more cells. So that's the disease part of it. And sometimes there's also they know that you're coming for them. They understand that the antibiotics. I'm sorry, the antibodies are being mobilized, and so they're going to attack. They're going to sense when you might do so. We learned about some of these responses before, and then when they sense that comfortable concentration using quorum sensing, they're going to elicit a toxic effect, right? Where they're going to release toxins to try to mitigate your response against them. Okay, so this is the typical kind of pathogenic steps. And so here we have a typical binding. Okay, so we have adherence. Let me put, all right. So we have adherence. So we've been learning about this with the SARS virus, right? Adherence is a really important concept, right? And adherence, ability of a microorganism. Boy, I saved so many letters there, didn't I? To attach to a cell or surface, because it could be like a tooth surface, right? So a tooth is not a cell, and you can have dental caries, right? And you can get disease in your gums and stuff. The first step would be attachment. So or surface includes those kind of surfaces, non-cells, but are still part of an organism, obviously. Biofilms can attach. So sometimes microbes can develop a biofilm, and that biofilm has then the ability. And the reason why that's important is because if you've established a biofilm, then you already have strength in numbers. So you come prepackaged, ready to go. All right. Microorganisms can they enter somehow through a portal of entry. Somehow. Okay. Puncture, wound. Usually something new that where they have access where they didn't before. Skin surface. Doesn't have to be dramatic. Bites. Something. And wounds enable microorganisms to access tissues normally inaccessible. So you have like staphylococcus, right? And I can show you like some skin-eating diseases that look like something out of a sci-fi film. It's crazy. You have microbes. Staph can like grow laterally and just start to take the skin, not touch the muscle, not touch the tendons. So it looks like you're like at one of those what's those exhibits? The bodies exhibit. Just takes the skin right off. It's so gross. Staphylococcus, what are you doing? You're supposed to be my friend. You're part of my normal flora. Why would you turn on me like that? Well, what happened is Staph, you had some kind of puncture wound or some event that now enabled Staph to have access to a different part of the tissue than it did. Now Staph can turn into a pathogen. So a lot of our pathogens live right on us. And they're okay as long as they're on our skin. But once you have a puncture wound, now you've infected the deeper tissues with microbes that were meant to reside on the outside. Totally different situation, totally different outcome. It doesn't always happen. I mean, we're not all going to drop dead, but that's typically how these things start. Okay, so we have adherence. All right, we have an adhesion. An adhesion is defined by a receptor on a pathogen surface that are specific to host cell membrane structures. Okay? Adhesion, important definition. Should have wrote that too. So here's a typical thing. You wouldn't be surprised by this, right? You have a bacteria. It has adhesion. We have a host receptor. They shake hands, and now the microbe is immobilized on the cell surface. Okay? So you have the adhesins all around the bacteria. You have the host receptor, they're shaking hands, and you have immobilization of the microbe on the cell surface. Here we have SARS-CoV-2. This is a very similar situation. Here we have that adhesins. The adhesins in this case are the spike proteins on the SARS virus, and the cell receptor is an ACE2 receptor. We talked about this, and in this case, what happens is the spike protein binds to the ACE2 receptor, and then this other protein we haven't talked a lot about. It's called a type 2 transmembrane serine protease. It basically cleaves ACE2. And this cleaving event activates the spike protein, basically cleaves into two, and then the leftover part of the spike then engages to open up this channel, and then the virus enters. So this is another example of adhesion molecules binding to cell receptors and then accessing inside. And now we have penetration, the COVID-2. All right, hold on. So I'm a little over. So I'm just going to give you one more definition before we start Thursday. We are finally at a very good part of this course that you guys have been probably hoping that this course would offer. Adhesins. It's just an important definition that I want you guys to have for the test. These are receptors on pathogen surface on the pathogen surface that are specific to the host cell membrane structures that are specific to the host cell membrane structures. And we'll stop with that. Yeah, that's a good stopping point. And then we'll carry on next time. And we'll talk about colonization, invasion. We'll go over the basic formation of a dental carie. It's going to be good. So with that, if you guys have any questions, let me know.
Dr. Cruz,
Yes.
so this Thursday is our last lecture.
Oh, is that a true statement? I don't want to looking through my thing. When did I say the final was? Let's see.
You said May 5th, but I think the GSU has on calendar this week is last classes.
Thursday is the last class.
Right, that's what I'm saying. This week, Thursday, yeah.
Right, and then
our exam is not until next week, Thursday, so we don't have a lecture on Tuesday next week, right?
I'd have to look. I thought that Tuesday was. I don't know when finals officially begin. They changed everything.
Usually the last class is on a Monday. Historically, that's been the case. I'll have to go take a look.
Well, I can take a look real quick if you guys are patient. I thought that Tuesday was available, but I might be wrong. Don't worry. This further justifies my speeding through all that stuff. All right, let me just take a look. Let me let's see. GSU final.
Yeah, Samara had a good point. I mean, if you have time and you would like to do a review session, something of that nature, kind of like a
I'm totally into it.
Okay.
Totally into it.
That'd be awesome.
Yeah. Right. Yeah, why not, right? Okay, we meet on Tuesdays and Thursdays virtually. Oh, wait a minute. This is I'm looking at the old one. So yesterday, hold on one second. So Tuesday and Thursday. Yep, yep. Actually, I mean, I think that I think that the 28th is the last day. I don't think Monday. I think 28th is. They have an exam starting on the 28th. Jesus, that's the last class for sure. Um, yeah. So Thursday will be the last class. Exams. So you're right, Mark. I think it was Mark. I wasn't looking. Um, Monday is the last class, so the exams, final exam starts on the 28th, and then we'll do it on the 5th. Hold on one second. Let me just make a note of that for my next class.
When I was gonna say, here's an idea as well, is that we could do some sort of video, just like we're doing like a class time, but it's technically review. And if we create a video of it, those that maybe have exams
That's a good idea.
have access, you know?
So let me see. Let me go back into participants. Everyone is mostly here. Oh yeah, packed crowd. I don't know who's sleeping or not, but so first question is, does anyone have an exam on on the 5th?
I think isn't everyone has like 3 days to take their final exam? So I think you have from May 3rd to the May 5th to take it.
Oh, I'm not doing that.
I know, but I think that everybody else, I mean, I guess maybe not everybody else, but most of us have 3 days to take the exam. So there's no like specific time that we can't take the
Oh,
exam.
I see what you're saying. So I'm the only bad guy. Um,
No, I didn't mean it like
no shade. I'm not going to go over any dental stuff now. I can't believe you said that.
that.
Um, no, I see your point. So I think, okay, I can't do a show of hands. Um, would it feel more convenient to have the final offered at a different time, like 3 p.m.? Or you guys want to keep it at 9:30?
No, a little bit later would be sweet because my sleeping schedule is
That'd be great.
gotten ruined since this COVID stuff.
Oh, Corey, where you been, buddy?
That makes me stay up late now.
Okay. So obviously this is a popular thing. I'm seeing like 3 or 4 yeses. 3 p.m. 3 p.m. Oh, jeez. Can't do 24 hours. Let's see. 3 p.m. So it'll be 3 p.m. I'm gonna make it a 2-hour exam. I don't know the format yet. What the hell does that mean, Dr. Cruz? What that means is that thinking about most of the exam, it's going to be cumulative, but I would like to add components for the extra credit that would be beneficial. And I'm thinking about, I don't know, I'm wondering what that'll look like, what the extra credit will be. Maybe it'll just be normal pathogen stuff, but we're starting to go over a lot of this microbial pathogen stuff. It just seems silly not to kind of cover some of this. Mark, I see none of you have young children. Um, it would be silly not to cover some of this SARS physiology stuff. So I'll go there. Let me see. Well, Mark, is what would be your preferred time? I mean, 9:30 can't be much better.
Oh, any time is good for me. I mean, if you want to do it at 6 a.m., I'm all right with that.
You'll be up. You'll be up. God, if somebody is avoiding medical school because they don't want to lose sleep, don't have kids. Medical school is only 4 years.
Absolutely, man. My biggest respect is the people that have multiple children that are young and are in med school. It is, I don't know how they do it.
Uh, yeah. Well, you mean are you an essential worker, Shanique? 3 p.m. Bad for your lab work. Okay. Well, then we could. Is 6
Hey,
p.m.
Dr. Cruz,
Yes.
here's the idea. Why don't you create some kind of poll that everybody can participate in and just put, like, I know they have this thing where you can pick, like, the time that works best for you.
Yeah.
Maybe you could do, like, a democratic thing or something. I don't know.
Well, I mean, yeah, that's I can't really leave anyone behind though. What about 6 p.m.? Is that better for everyone? Is there anyone? I mean, I want you guys to speak up. Don't feel like I'm just trying to. So Amy wasn't for the other one. That would be lovely. Okay, so now I'm getting love from that. How about the other one? 6 p.m. Look at that. Emphatic. Okay. Yeah, 2 hours to complete the text. 6 hours. The problem with that is the whole sharing of exams, and that gets tricky. Not that I don't trust you guys, but just imagine if you're honest and other people aren't, and then they start to mobilize the grades upward, and then it takes away the curve, where it gets really weird. So I'm trying to construct the exam so that you kind of have no choice but to take the exam from studying. The other exam scores from exam 2 convinced me that was a good strategy for keeping people honest. Wasn't really, I mean, the exam scores were actually pretty good. I had around 75% average and stuff like that. But the format of grabbing questions from a 100 question bank, I learned some lessons from that. No redundancy, all these things. But I think if I have like 100 questions and then 20 questions, that worked out well. And then you only see 4 questions at a time. You can't go backwards. What I want is to force you guys to be prepared for the exams. I can't control anything else. Sad I want an A. Why not noon? Everyone should be up then. That becomes tricky for the day people. That's why. People that do have a job or anything like that. That's why I said 6 o'clock. So I'll talk to my colleagues and see how that other open time period works for them. But for the most part, I think that it'll be a 6 p.m. Test. I think that's pretty good.
Dr. Cruz.
Yes.
So,
Do you?
sorry.
Yeah, go ahead.
Okay. Are you planning on doing 100 questions and shuffle that like the last midterm? Because we've got like questions only from HFR. That was not very cool, I think.
Yeah, I mean you got to know everything. And so
My
you
exam
can't
was over
really.
HFR.
Yeah, but we studied so of those mechanism and all that viruses and everything. And you know, HFR also we did, but we put on so much time to other things too. Didn't show up at only HFR from so many questions, like, and we didn't know everything, so that made us fail. So I don't think that was very
Why would it
fair.
make you fail? Give me all the same test bank, but the
Mostly because like Niha, we only went over about 20 minutes of conjugation with Niha. So when my entire exam was
Yeah,
HFR, I really didn't have
mine
any
too.
idea what was going on. But then when I saw all the exam questions
First
that
of all,
were
I
in,
didn't make the exam.
like, holy crap, I got
First of all, I didn't make the exam. I only organized it.
right.
So there's that. Um,
But yeah, like seeing all the questions
so then what I'll do is this. This is what I'll do. I'll create multiple test banks, and then I'll have it randomly take from these different test banks, right? So, like maybe I'll say 10 questions from, I don't know, what did we just cover? Microbial adherence or whatever. Then I'll have 10 questions on microbial adherence, and then your exam will have 3 of those 10 questions. So it'll kind of stratified according to topic.
Would it be possible to just have everybody have the same questions, but it just go in different order and you just can't go back to another question? Because like when I
It
saw
doesn't.
like all the test bank questions there were like all 100, I was like, man, I kind of got the hard side of this exam compared to what other people could have gotten.
Oh, you didn't see everyone's score.
I'm sure, but like my entire exam was HFR, and I spent like 4 or 5 days studying all the other stuff as well.
Yeah,
And
exactly. That's what
it was only
happened to me
three
too.
Kind of.
hard.
I think that is always the case, though. I mean, I think I could stratify the topics to make it a little more fair, but I think that's just the way it goes. I mean, you can't always, even if I made all the questions organized, it could have still been the same. I mean, it's just the luck of the draw. I mean, I've just been through this, and I feel you, and I don't like. I don't know. It's just I understand. I understand how you feel. I understand how you feel. You want to study that which is tested. Um, can't always control that, but I could try to do a better job. I'll stratify the topics and then draw from different pools, and so that it doesn't have a bias towards picking questions that are related when it doesn't know the difference. Now it'll know the difference. And that's pretty
Okay,
good
awesome.
feedback.
Yeah, that'd be really good.
That's pretty good feedback. That's really good feedback.
The pack back questions, is this the last week for pack back? And what's the, do we need to know like what's the total of questions and answers we should have by now, or it's week by
Well,
week?
I'll have to look at it because it's a little tricky, isn't it?
Right, right. The first week that pack back was not, we hadn't done it yet, so that week is empty. So I just want to make
Oh,
sure.
it's pretty forgiving. If you had me for the fall for 3,800, you'll realize that if you've been mainly participating it, I only ask for you to be really a part of 80% of it for you to get it, all those points.
Got you. Got
Is
you.
it going to be graded the same way as last semester's as well?
Yes.
Okay.
Um, all I ask of all of you guys is that you guys try. The semester went sideways. There was a lot of things that, and I'm glad you guys still have an enthusiastic attitude. You guys are very well mannered and professional, and I appreciate it. I think that no matter what happens, you should always adjust. I don't think anyone should be clamoring for excuses on anything in life. In this class, I haven't heard a lot of that. My other classes, however, um, and I think that you can't predict what's going to happen. I don't think you can control things. I think that adjustment and evolving is the way to be. But you know, I also understand that you guys are trying to build your lives and careers, and this is a little bit disruptive. Minorly, um, you know, sympathetic towards that in the sense that, mostly everyone has been doing okay. The exam two went, but I'm not trying to fail anybody. I'm not trying to screw you over. I'm not trying to make this hurt. I just need your best effort. Best effort. I will be calculating everyone's grade at the end. I can't turn a 42 into an A. I can do better than just having you have been set back by this. I think we can do a lot better. So, um,
So, Dr. Cruz, are you saying that if someone has like an 86, that we're gonna get an A?
I don't know
I mean,
about
I'm
that.
not saying that I have an
Like
86.
it,
I'm just
let me
asking.
tell you how the math works out usually with this grading scale, just in general. The way the grading scale exists currently is that, and and it's amazing. It's just fortuitous that it works out that way. So there's some things that I've not seen violated in the 14 years I've been teaching. I've never seen someone get a grade in a class that they didn't get on an exam at least once, right? So it's hard to get an A in a class you didn't get an A on an exam. That seems fair. Second thing I've never, and so then also if you get if you get over 80 to 90% on your quizzes average and your uh, uh, combined quizzes and backpack, if that is over like 80%, then if you're at 85% on your exams or 75%, or let's just say 75%, say if you have a 75% on your exams, then what happens is the way it works out is that you'll be getting that next letter grade. So it's about half a letter grade worth this backpack and the quizzes, which is quite a lot. And so, yeah.
And so you're still dropping the 2 lowest quizzes?
Still dropping the 2 lowest quizzes.
Will we
And
have a chance to retake? Oh, go ahead. Sorry.
mhm, um,
Will you offer like any additional extra credit, please?
there'll be quite a bit of extra credit on the final.
Thank you.
Thank you
That's,
for that. I appreciate it because this whole online stuff with all of our classes has been miserable.
oh, is that right? I'm sorry to hear that, Cade.
Gosh, it's just so much work. It's insane the course load I'm under.
Yeah, true.
I have 4 reports due on Friday, a group presentation, 2 exams on Thursday, another 2 reports due that Monday, and these are all like 12, 13 pages each. So it's like, it's crazy.
What classes are you taking, if you don't mind me asking?
Spectroscopy, chromatography, molecular cell biology lab, and then this class and my research with quantum physics.
Jesus.
Yeah,
Hey,
I'm getting
hey, man, at least you're in school during this
I
time.
mean, I'm doing really good in all of them. It's just like
No, but it's a lot.
11 to 9 every day.
Oh, I know. I mean, I remember, I think it was the semester, I think I was taking cell and molecular biology, microbiology. I was in undergrad. I was in um, University of Florida. I'm generation educated, so I'm like just scared to, I don't know anything. I feel like I had imposter syndrome, and for some reason, the way that semester worked out for me, I was taking an exam every week of the semester, and then that also interrelated with papers and stuff. I was just working so much that I just had to like just think that psychologically, and it's just there's an end date, right? There's a final date. This will end at some point. Just don't ever stop working. That's all I remember. And I remember at the after I took the last final, after all of it, I remember it was a microbiology final. I like walked out. I had been so shell shocked by the whipping I had been taking all semester. I remember when I got in my car, I just put my head on my steering wheel. I remember just crying. I wasn't upset. I don't even know why. I just can't believe it. I'm like, oh my God, what the hell? Like, so I understand you, Kate. It's tough, man.
I totally agree with that feeling. Sometimes you just want to like tear your hair out. You're like, oh my God, I don't know how to do all
What's gonna
this.
happen next? Samara's crying
Get
now.
through it, and once it's done, you feel really good about it.
Yeah, it's just a release of energy. You're
Yeah.
like, whoa, yeah. Um, how do you have a group presentation via the computer? That's insane. Yeah,
Hey,
yeah.
Dr. Cruz,
Yes.
like my internet was cut off, but will we have any study guide,
No,
um,
not for
for
this.
the
Um,
for
this is just the
the
stuff since the exam two. Um, I think that the only thing we have on this chapter is chapter 14. Well, no, that's not true. This is chapter 20, 23, some of 14, 24, 25, maybe 26, mostly 25 though. I can create something if you want. I can make it nice like that.
Yeah, please, that'll be
Because I'm
really
gonna have to create organization anyway because of the points that Kate brought out. All right. Study guide.
or practice questions.
Oh, that's a good question. Let me put that in my notes too. Yes, practice exam.
Yes.
Practice questions, I should say.
Can you also do like a Kahoot or something? And like
Again?
if we get, could you also maybe do like a Kahoot? And if we get like so many correct, we can have like an extra credit or something? I don't know.
We can do a Kahoot. We'll do study session or review session. That'll be so fun, actually. So that'll be part of our review session is we'll do a Kahoot. And we could do that next Tuesday and Thursday. Do 2 review sessions. There should be no reason why you're not prepared for this exam. I mean, this is going to be, I'm going to give you the information. I just want you to earn I don't like curving. I want you to earn all of it. But that's really cool. So next Tuesday and Thursday we have for a review session. So we have quite a bit. We have a lot of opportunities to kind of go through this, and for you to have exposure to a lot of the questions. It's really good. Okay, so I'll put a review session, and because the reason why it's easy to make a study guide is because I'm going to categorize everything anyway on the exam. So I have to think about what different topics the question pool is going to come from. So,
So if we're gonna like create a study guide, like us by ourselves before you do it, would it make sense to like go through the book and just like kind of look at the chapters that we went over and just take the main topics from
oh my
there?
God. All right.
Or slide.
No, it wouldn't make sense.
Okay.
Let me see. I feel a little bad that it's 11:15 and people may have had to log off. I do, I think I stopped the material and I'm recording this and I'm offering it. We're just basically rationalizing, leaving people behind. Let's just go through the different things that are going to be on it, since you're obviously eager to kind of go forward. So we're going to go chapter 20. There's all those definitions: ecosystem, population, community, species richness, species abundance. And then we're going to, we define biogeochemistry. Then I want you to understand definitions like biofilms, microenvironment, microbial mats, right? And then that's going to take care of chapter 20. Then chapter 23 comes up, all the common terms for all kinds of symbiosis. Then we have example symbiosis. This is where chapter 3 becomes a little bit easier to kind of consolidate. So you want to focus on lichens. Then you want to focus on consortium. The example of a consortium that we went over is Chlorochromatium aggregatum. You want to know about all of that. And then you basically have um, the plant nodules, right? And we talked a little bit about mycorrhizae, but that's very minor. Most of it is going to be plant nodules. So that kind of cleanly cuts chapter 23 into three main components. All the definitions, symbiosis, then lichens, consortia, and plant root nodules. When you go on, I went on to chapter 14, you'll have to know the photosynthetic pathways. That's really what I care about. And it's not that hard. You just have to get through it. I want you to be able to distinguish between anoxic and oxygenic phototrophic bacteria. Okay. So what are the differences? What are the electron donors is basically what I'm asking there.
Dr. Cruz, sorry to cut you off, but that
Yes.
photosynthetic pathway, that's 14 point, what is that? 3?
I would say for figure 1414.
14, 14, okay.
That's the main Z scheme. But that is packed with a lot of concepts because you have a photosystem. The photosystem is basically the light harvesting um, antenna molecules that are directing it to a reactive photocenter. So you have to know all that. But yeah, if you know the Z scheme, you understand it, you'll be pretty good. And then you know the difference between the schemes between. You have to basically, anoxic bacteria have a cyclic light dependent reaction, and then oxygenic photosynthesis has non-cyclic. You have to understand the difference between those. But then otherwise, I went over some autotrophic things today, and then chapter 25 is coming, and we haven't gone over all that. That's why it's hard to tell you right away. But that'll be it, chapter 25. So that's the basic guide. So if you were to go forward, what I would do is I would go through your chapter 20 and chapter 23. Those are really good places. That's really good. That's what I would focus on. And then you can go for photosynthesis and stuff. But that's what I would focus on. That's a good use of your time. There's still 34. Still 34 what? 34 people?
People like hearing the ending questions.
Oh, I'm not sure. Whoa, 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 whoa. I'm not sure if we said this already, but there will be more quizzes next week before the exam. Yes. People want quizzes because they want to fade out. They don't want their drops to be used. Yeah. So quiz on chapter 25. I'm going to be talking more about 25 on Thursday. Okay. So there'll be more quizzes. Drop 3 quizzes. I'll drop 3 instead of 2, just because I'm worried that some folks out there had inconveniences with resources outside of the university. I know people were kicked out of their dorms and all the good Wi-Fi. So, all right, guys. I do care. I'm trying my best. Oh, Neha's quiz. I think she's going to put it in. I just talked to her last night. She's going to put it in. She didn't want to put it in without telling me. But yes, I'm going to put hers in. It should be, well, I'm not doing it. She's doing it. So, and I'll be giving a evaluation for her. And, it all goes around, right? I mean, let's help each other out. I'm sure that you guys will view her favorably. And anything that she didn't do right, we're all learning, and I know she'll be just excellent. And so I hope that you guys favorably review her. I can't obviously tell you what to do. And I appreciate your guys' patience with all of this too. And I'm going to try to give you all the opportunities to make a good grade in this class. There's no reason why you can't do it. I really don't think so. I think that as long as you work, that's all I'm asking for is for your work. It's not a lot to ask. Everything should be okay. A's. Everyone gets A's, right?
I have a quick question about the quiz root nodules. And I'm just so for my own knowledge, because I second guessed myself, which was, what benefits are conferred to rhizobial species inside a root nodule. And perhaps I got it conflated with leg hemoglobin, because I originally put access to sugars, which is, from organic acids. But then I switched it to anaerobic conditions because I was thinking, well, with leg hemoglobin, being, what it is, I mean, it's there for a reason, that I was like, well, maybe it's the anaerobic conditions and, versus.
The leg hemoglobin's inside of the bacterial cell, so it's not really a benefit from living with the plant.
Okay, so it's inside the cell, so it doesn't necessarily create an anaerobic condition for the and the path. That's not a pathogen for the I can't even pronounce it. Rhizobia.
Well, let me just, yeah, the rhizobial species has to, because it's forced to be in an aerobic environment, it's forced to deal with has to evolve a mechanism to maintain tolerance of aerobic conditions and output at, see, because, okay, let me just review here. Leg hemoglobin is inside the rhizobial species. That's a short answer. So it's not something that the plant does. The reason why leg hemoglobin has evolved is because if the bacteria is going to be living with a plant, the plant's cranking. It's growing fast. So the bacteria has to be able to deliver nitrogen at a rate that the plant benefits from. So it's got to crank up its metabolic speed. And it's doing so in anaerobic conditions because of leg hemoglobin. So leg hemoglobin just makes it so the rhizobial species is not hurt by all the oxygen being evolved by the plant.
I see what you're saying. So it's basically, it's just trying to pull off any breaks or any impediments or roadblocks. It's not necessarily a benefit to the rhizobial species.
Rhizobial species is where the leg hemoglobin comes from. So it's not something that the plant does for it. What leg hemoglobin does is it basically offers oxygen as an electron acceptor to the electron transport chain.
I gotcha. Okay, I think that's what I did. I think either the way I wrote it down, I must have thought that it was coming from the plant versus the rhizobial. Okay, that's where I made a mistake.
Yeah, Dr. Cruz, I have some question about the quiz too. The lichen that has algae on it, and the question was which one is not fitted the algae, and I thought algae is supposed to photosynthesize, and access of sunlight is supposed to help the algae, but it's
But the
not
fungus
benefit.
doesn't really help sunlight become more or less available. Um, Sunlight's there regardless. Like the fungus isn't helping the algae gain access to sun. Sunlight's there no matter what. What it is doing is it's staying moist. It's excreting enzymes that are digging into rocks and barks that liberate minerals and nutrients that the algae can then acquire. It's mostly these inorganic materials that are limiting to an algae. So that's the real benefit. I think you might have been considered this would, but when you're thinking about a consortia, maybe you were thinking about a consortia. A consortia is a freshwater symbiosis where we have like green sulfur bacteria living on a motile bacteria. So then in that case, the the motile bacteria, the flagellated bacteria, is going to be needing to go towards sunlight so that the motile bacteria, so that the green sulfur bacteria can photosynthesize, but it needs to keep it outside, but not near any oxygen. So that, in that case, sunlight would be a good answer. But algae doesn't really get sunlight from the fungus, and the fungus doesn't move around towards sunlight. It doesn't. Yeah, that's a good reason why to think about it. Fungus doesn't move.
Also, I have a question.
Yeah, what's up?
So you said that for exam two, if we didn't get the I love microbiology question, that we would get a +4 for it.
Yeah.
If
Which
that
one?
+4, I love microbiology question,
Oh,
if we did get it, you said that we would get it if it is like the
it's
influence of
there.
our grade being on the next grade scale. How will we know if we need that 4 points or not? Will the grades be posted before they're on deadline day,
Oh,
or
it's yes, of course. You will. I see. I see. Remember. I know what you're talking about now. It's simple. The grading scale I think for this course is kind of on a points basis, right? So if you have 440 points, you get an A or something like that. So you just add up your points, and if you're 4 points shy, that question mattered. Pretty simple.
so for exam two, you said that you were gonna add points, or maybe there's a curve or something, or is the grade that we have now the total grade that we have for the exam?
I am. Oh, I have not addressed that, have I?
Uh,
So for exam two, I will put a 10-point curve. Because I'm not grading these physically, I just kind of forgot about it. All right, so I'm writing that down now. So exam two, 10-point curve. Dropping 3 quizzes. I better stop this session. I'm just giving away the. All right. Yeah, I'll put a 10-point curve on there.
Awesome. Thank you so much, Dr. Cruz, for being so forgiving about it.
Yeah, of course. 73 points. Well,
Dr. Cruz, we
it's
also
hard
have
to
that
it's hard to get a good
gene
grade.
mapping question, right?
Say again?
We also have that gene mapping question, right?
Should be fixed. If it's not, notify me. There's a couple people that have notified me and said, hey, this one question didn't get corrected, and I've gone back and corrected it. But I've definitely gone back through every exam and corrected it. Check your points, though. Thank you.
I did, but it didn't make sense to me actually, because I did everything correctly on the practice exam for the pretty much the same question, and just that the question read out didn't make sense to me when I chose the answer in the exam. I had to choose one regardless the answer. So yeah, I don't know how you
You'll
did
just have
that.
to email me specifically, give me a screenshot, and then to explain your thinking.
Okay,
what
we'll
a
do.
lot of people have been
Thank
doing.
you.
Yeah.
Yeah, we'll do that,
So you
Dr.
typically with the way people have been handling this, because that question just something was wrong with the test bank. So the way I've been handling it, so people have been just screenshotting it and then telling me, oh, hey, I didn't get all my points, what's going on? And I'm like, oh, I didn't see that, or they'll explain something to me. But for the most part, I went through and everything. So please, though,
Yeah, I'll
email me
just
to
I'll just
make
send
it
you
individual.
shot and explain what I thought when I picked the answers.
Yeah. All right, guys. Okay, good. All right. So then I have all my notes here for all the verbal contracts I've delivered. Jeez. Yeah, my daughter's school, I don't know why they would send this email out. Send an email to all the students saying, well, your grades won't go down. These kids won't get up before noon now. I mean, why would you send that email? Oh my God. So I want you guys to work a little harder than that. Exam 1 grades. Ooh, that's true. I got them all. Yeah, got them all right here. Yes, I should put that in there on my to-do list for this class. Man, you guys are keeping me busy. All right. Post. And 1. Okay, guys. I got to go now. Encroaching on my next class. So, okay, guys. I'll see you on Thursday.
Dr. Cruz,
Yes.
I think we have you wanted to go over this presentation
Yes.
slide. Okay?
So everyone, yeah, that's the next thing I have to go. So everyone else kind of log out, and then I'm going to be with Unu, and then we have to discuss some things that she's going to be doing Thursday. So I appreciate you guys. Take care.
Thank you.
Good one, Dr. Cruz.
All right, take care. Thank you. Unu, why don't you go ahead and share that screen?
Okay. So, can I email the slides again? For some reason, I can't. My computer doesn't let me pull up. I can't share a slide from my computer, so I can email the
You know how
presentation.
to do it?
Okay, but I do have the slides, so I can kind of go over it if you still
Yeah.
have the
Do you
one I
know?
sent.
Do you know how to do it though?
I'm using my phone. Let me see.
You're using your phone?
Yeah, for the web.
Oh, no. Don't do that. Email me. Yeah.
Okay.
Are you sending me an email now?
Yes, I will.
Okay. So then when we'll present it, I'll host the PowerPoint. You just talk me through it.
Please. See here. Oops. Let's see here.
Let me know when it's done.
Okay.
Just doing other things until then.
Okay, Dr. Cruz, I just sent it.
Hold on 1 second.
Okay.
You got your schoolwork going? Okay. All right, I'm back. Did you get it to me?
I did.
Okay, all right. Okay, sorry. Get into my email and then it's just I get so distracted. Okay, so you got the presentation. I don't see a presentation here though.
It's going to be the most recent one.
Okay, I see it. I gotcha. All right. Man, you're just sliding right in here, right? You're just in time, aren't you? Just getting you're getting this presentation in just on time. It's this is the time to do it. I mean, you see what we're covering now, so it's appropriate. It's totally appropriate.
Yeah,
All right,
very.
you go ahead, and you let me know when to click.
Okay. So so I'm just going to go ahead and introduce what my topic is about, which is discussing the microbe and the disease that is attached to it, which is malaria, to Paramecium falciparum. And then what I'm going to state on the next slide will just be the facts associated with malaria. So it's just going to be a summary of malaria. Okay. No, I think that's the wrong presentation. That's the first one, but I had to change that. So
This is the one that. All right.
yeah, that's the very first one which I changed. So it's the most recent
Well, this is
email I
you just
sent.
sent this to me at 11:32, so I think this is the one you just sent me.
Yeah, the most recent one. It's taking a little while to go from here.
Oh, I see. 11:34. Yeah, I mean, I think that's the one I have. 11. Do you see my presentation? Or you see my screen, don't you?
Yeah, I see your screen.
So this is not it?
No.
You sent me the wrong one, I think.
Send it. Dr. Cruz, I sent it again. I'm sorry it's taking so long to get it out.
All right, let's take a look.
Okay.
Is this it?
Yes, that's the one.
Let me download it. How to I don't know. We'll try this. All right, let's go. I don't know how to make this present. I'm not used to using this. Wait, present. All right, with me?
Yes, that's
All right. So
it.
you told me about that, and then we'll go to the next line, Lateral facts.
Okay. So did you want me to present like I was talking to the class or just sharing what I've
Yeah,
done?
just do it like you're talking to class.
Okay. So good morning, class. My name is Unu. I'm going to be presenting on a very important topic that I feel is important to me, which is malaria. Looking at the microbes, Falciparum falciparum, which is the causative pathogen that is responsible for the disease. So on the next slide, on the first slide, just show some of the facts about the malaria disease. It's responsible for over 450,000 deaths per year. Typically, it's caused by two popular strains, the falciparum and the vivax strains. The distinct thing about malaria is that it has two hosts, which is the human and the female mosquito host. Symptoms include high fevers, acute kidney failure, death in most instances if it's not treated. And then we've also seen that sickle cell, red blood cell carriers have a resistance to malaria disease. Our next slide shows the structure of the microbe. So basically, it's an amorphous protozoan. So that means that the morphology keeps changing depending on which host is the microbe is attached to. With the liver cells, it takes on the form of the eukinides and the sporozoites. And when it's in the red blood cells or the malaria mosquito, it's in form of the gametophytes. And then there's also the apical complex vital, which is vital for the mobility and adhesion to the red blood cells, and which is something that we discussed in the lecture that we had today when we looked at how microbes are able to attach to a particular organism or a particular structure that helps it stay or abide within a host and makes it capable of causing diseases. So the same thing happens with Plasmodium. So the apical complex is a very important structure for it in adhesing to the red blood cells. The life cycle of, which is the next slide. The life cycle of malaria consists of two hosts. So this is a pretty simplified structure.
Like
it's actually,
much
yeah,
better.
it's
Much
much
better.
better. It's much more complex than this, but just for the purpose of the class, it's been broken down into 4 distinct cycles. So you have the first stage, which is the infection. That's when the mosquito bite occurs. It forms the sporozoites, which get multiplied in the liver cells. So within the liver, there are actually 2 ways. It can go on to cause diseases or just stay within the liver and not cause any disease. From there, the next part that it goes to will be the red blood cells. So that's the most disease-causing stage. It forms the merozytes. Within the merozytes in the red blood cells, there's a asexual cell cycle. So that's how it duplicates, forms the gametocytes, and then the gametocytes go on to the sexual reproduction stage in the mosquito. So as far as treatments for malaria, for the preventive anti-malaria quinine drugs, usually targets the stages A and B. So at that stage, the cells are simply infecting and proliferating, and they are still, they are able to, the drugs can work effectively. But by the time it goes into the red blood cells, that's the most dangerous part. It's usually when it goes on to cause death in most instances. Then the next slide shows the red blood cells mutations and its relationship with malaria. So with sickle cell is a disease that is, it's a genetic disease caused by mutations of the HBB gene. The most important thing to remember about sickle cell is that it's a missense mutation, meaning that one of the proteins is changed completely from valine to glutamic acid. So because of that single change, it also changes the structure of the red blood cells from a regular rounded normal red blood cell to a sickle cell shaped. And they're just, the pictures I have showing here are just 3 different cross sections of what a sickle cell shaped blood cell and a normal blood cell shape would look like within the capillaries. So the pictures here are actually that of someone who's suffering from sickle cell. The symptoms for sickle cell is usually intense pain. In a lot of instances, it does cause death, and then there's a paleness of skin and jaundiced eyes. So you see the sickle cell blood shape and the rounded hemoglobin, which is a normal blood shape, both existing within a human who is pretty much a carrier of sickle cell. And the next slide.
So the disease is homozygous or heterozygous? When you say that,
It's
if you have homozygous, then you're dead. Okay, never mind.
Yeah. So the carriers are heterozygous.
Yeah, so you can't be a homozygous. You'll just be dead.
Yes. So the slide here tries to explain what could be responsible for why sickle cell carriers don't have, or I mean, they will be bitten by mosquitoes because most of that occurs in the tropical areas, but they don't go on to develop the full disease. Has to do with the sickle cell nature of the red blood cells and the fact that in sickle cell carriers, the phagocytes, or there's the process of phagocytosis, which means that it treats the sickle cell blood cell that is carrying a mosquito or a plasmodium microbe as an invasion, and it will phagocytize it, meaning it eats it up and then disposes of it. But in the normal blood cell, that's not what happens. So usually they are able to evade that stage and then go on to the red blood cell to cause full-blown diseases.
Okay. I think that on this particular slide, you should work on, I think, first of all, anytime you present, if you put a poster, you start with your knowns and then you go to your unknowns. You never go the other way around.
Okay.
If you make a poster, you start with your things that you know on the left-hand column and ask because you read from the left to right. So then you go to the things you know and then to the things you don't know. And so here you want to start, and then we go from left to right and from top to bottom. And so people aren't going to start with the bottom and go to the top, but it's not. This is appropriately organized because you have the normal on the top anyway. And so what I would do is just talk about this in terms of what happens normally. So you just say normally when you have an infection, you go through proliferation inside of the red blood cell, and this red blood cell is not able to be recognized by the phagocytes, and the microbe is able to continue and proliferate and continue its life cycle through the red blood cells, through the liver, without detection, because it's able to continuously coat. But when this in a sickle cell carrier, a lot of the red blood cells are not a hospitable form for this. What do you call it? Plasmodium. And so, in this sickle cell state, I wish this was a sickle cell. Anyway, in the sickle cell shape, the microbe cannot thrive, cannot go through its life cycle. This is a structure that's recognized by the um, phagocytes, and the sickle cell red blood cell is taken in by phagocytosis. So just very cleanly just state what happens and then what doesn't happen.
Okay, so kind of work from the top to the bottom in explaining.
Yes.
Okay.
And it's a teachable moment here. You did do a good job saying it. Maybe I'll tell them afterwards if you don't, but we have one single nucleotide that resulted in one amino acid difference in a protein, and just changing this one amino acid disrupts the entire shape of the protein such that the entire cell has a different shape.
Mm-hmm.
That's remarkable. It's easy to think, oh, just one nucleotide. Boy, that has big consequences physiologically.
Yes.
It doesn't always, and it usually doesn't, but here it does. Okay, I'll let you do the conclusion.
So the conclusion typically and is just to kind of think around how, and tying it to what's currently going on in the world right now, which is possible treatments using anti-malaria drugs for coronavirus, which is a big thing with us right now. So some of the drugs have been used have been anti-malaria medication like chloroquine and hydroxyquinone. So the question for me, and maybe for a lot of people who have maybe in the past been exposed to anti-malaria medication, could it be that there's a reduced immunity that along the line you've developed by the exposure to this medication? Could it also result that, since it's an anti-malaria, is it a preventive medicine or it's just used for treatment, and could asymptomatic carriers that are exposed to this drug, does that, confer on them some type of immunity? And how does, the plasma of someone who's suffered from malaria and being treated with this anti-malaria drugs, is it something that, in the future there's a potential for vaccines from such people? So those are some of the things that I just kind of pondered about, and it might be something that,
It's relevant current research. That's good. That's good. You did a good job.
Thank you.
Yeah, I think that you did a good job. I like what you did. You simplified it. You made it accessible. I think that students will get a lot out of this.
Okay. And we're still looking at a 10-minute presentation time. Yeah, say kind of like keep it like 10 minutes.
Yeah, I think you're there. I think that
Okay.
I think you're there. I think this is great. I like the yeah, I think it's great. Um, Let me work on my so next time what I'll do is I'm going to be going over some I'll cue you when it's time for you to present. I don't want you to do it right away.
Okay. Will it be like in the next class or the upper week?
Class. It's definitely next
Small
class.
class. Okay.
It's the next class. And
I'll be
it'll
ready.
be, I just want to cover, I'm going to do adherence, and then when I cover virulence and the disease causing stuff, that's when I'm going to cue you in. It'll probably be about a half hour into the lecture.
Okay, cool.
All right. So I'll have
Right.
the presentation ready to go, and then you'll be on standby, and then I'll just walk them through it just like that.
All right, awesome.
Anyway, that's good work. I appreciate your work. You made
Thank
this
you.
much better.
Thank you so much. It's an area I'm really kind of very interested in, and hopefully, just to see if something comes out of, anti-malaria drugs. And because
Yeah.
I lived in Africa for a while, so I've been exposed to things like this. I don't know how, it's going to affect me or other people who have been from the tropics and exposure to coronavirus and the medication. So it's just something that I've been very curious about.
Yeah, I mean I'm not sure what the the rates are in Africa, Zimbabwe and stuff, but it just seems like the cases are a lot lower. Now it could be due to
Yeah.
testing,
So,
but
some say it might be the heat, you know. So I mean, I try to like,
Florida is hot and Florida's not necessarily doing great, so I don't think that's it.
mhm.
Could be some of the situations that immunities to other immune attacking microbes that offer some advantage. Maybe Africa will take over all those countries. Anyway, really good work, and I'll see you on Thursday.
All right. Thank you, Dr. Cruz.
Of course, take care.
All right. Bye bye.